Hello, everyone. So today I'm following on from Brent's talk just now, which explored the methodology behind using RNA-seq across parts of the avian brain relating to social behaviour. And today I'm going to tell you about my ongoing work in this space and about how gene expression in the brain relates to male-male cooperative behaviours in the white-tailed manakin. As again, this is a massive collaborative effort, and without these collaborators, I would not know my amygdala from my elbow, so big shout out to these guys. So in birds, social behaviour is best characterised with respect to male aggression, paternal care, and male sexual behaviour. And we also know that these behaviours are modulated, at least in part, by the hormone testosterone. So testosterone modulates social behaviour through its regulation in the hypothalamic pituitary gonadal axis and its action through the androgen signalling pathways within the social behaviour network. Within the social behaviour network, there are multiple ways testosterone can act through different channels of the androgen signalling pathway, as well as through multiple nodes of the social behaviour network, which Brent has just described. These nodes of the social behaviour network are defined by different densities of hormone receptors, steroid-sensitive neuropeptides, and steroidogenic enzymes. However, these nodes are thought not to act in isolation, but in concert, showing differential activation of various nodes to produce different aspects of social behaviour in a landscape of activation. So most of what we understand about social behaviour and its neuroendocrinology comes from Northern Hemisphere passerine birds with highly seasonal breeding and socially monogamous breeding systems. So not only do we have not a good representation of the diversity of avian social behaviours, these studies have also tended to be fairly simple in scope in terms of tissue resolution and the number of genes sampled. So this study on the white-tailed manakins goes against this grain by taking a holistic approach and looking at the underpinnings of an unusual male-male cooperative behaviour. So in the white-tailed manakins, territory-holding males will perform cooperative displays with other, usually subordinate or floater males. The more cooperative territory holders are more likely to gain paternity, and the cooperative floaters are more likely to ascend to territorial status later in life. So there is this fitness benefit to cooperation. And previous work has shown that testosterone modulates individual in like attendance and cooperation in a status-specific manner. So there's a whole laundry list of questions that we want to ask with this data set, but it's an ongoing project, so today I'm only going to focus on a few relatively simple ones. So at the very basic level, we are interested in what genes are involved in these male-male cooperative behaviours in brain and endocrine tissues, and how patterns of gene expression overall differ across these neuroendocrine tissues and associated with different aspects of the male-male cooperative phenotype. So to answer these questions, we sampled 12 territorial and floater males from Tipitini Biodiversity Station in Ecuador. These males were sampled over multiple weeks and seasons with repeated behavioural and hormonal observations, including circulating testosterone, which has already been demonstrated as a modulator of behaviour in this species. They've been characterised with respect to lek effort, which is a measure of time spent at a display site. Social network strength. This is a metric which describes how often a focal male will associate with other males in his network. And so the thickness between the nodes represents how often they associate. And social network degree. And this is a measure of the number of unique partners a focal bird has. And as Brent has already mentioned, we expanded the tissues sampled previously and to encompass a wider variety of tissues that are thought to be important. And most notably, that Brent didn't mention, we also included these endocrine tissues of the pituitary and the gonads. So in total, we sampled 12 tissues across these 12 birds with detailed behavioural data. So I'm not going to go into detail here about the methods, but for each of our 12 tissues, we have RNA sequencing data, and after filtering out we have expression profiles for over 16,000 genes in our 12 brain and endocrine tissues. So one of the challenges of these highly multidimensional data is to try and pull out meaningful patterns whilst reducing some of this dimensionality in order for the human brain to comprehend it. And so one of our predictions is that we will see patterns of gene expression change in each of the nodes with respect to the behaviours of interest. The model presented here is based on expression patterns of a few key immediate early genes, so genes that are the first responders to a particular signal. These gene expression patterns differ with respect to the different behaviours displayed. So we wondered if these different landscapes of expression can be expanded to our aggregate gene expression profiles across our tissues. So to look at this, we ran models for each of our traits and each of our tissues and looked at the number of genes that were differentially expressed. So each graph here is the results of the differential expression analysis for each of our traits. So we have status, lec effort, testosterone traits, and our social network traits. On the x-axis, we have our individual tissues measured with gonads on the far left. And as we go right, we ascend upwards through the brain to get to the bed nucleus of the stria terminalis on the far left. So each of these bars represents a tissue, and the dark coloration below 0 represents the number of genes that are identified as being downregulated with respect to that trait, and the lighter color represents the number of genes that were upregulated. So broadly, you can see here that there are different patterns of gene expression across the different nodes of the social behavior network and the other neuroendocrine tissues. So we're confirming this prediction from 20 years ago based on the responses from immediate early genes, and show that aggregate patterns of gene expression in the network will act differentially according to the hormonal signal or the behavioral phenotype. There are also some other interesting things that this plot shows. The first thing is that we have quite distinct expression profiles according to our measure of testosterone. So our understanding of the relationships between behavior and testosterone are based on the mean testosterone profile of individuals over the course of the study. However, we also know that individual testosterone levels vary through time, and that a change in a signal will also produce a change in gene expression. And that's exactly what we see here. So the testosterone levels at sacrifice have a different gene expression profile from the aggregate mean gene expression. And we also, we also find that there's a huge number of genes differentially expressed in the bed nucleus of the stria terminalis with respect to lec effort. So just to point out that this y-axis here is quite different from all the others, and so we have over 2,000 genes that are differentially expressed here. Now, this nucleus is at the intersection of the social behavior network and the mesolimbic reward system. And so this is lending suggestive that maybe this is a really important hub of activity in this species for courtship behavior. And so we have all of these expression profiles, but what are the genes involved? Our other question was asking what genes? And so one way to get at this question is to look at genes that we think a priori will be associated with social behavior. So knowing what we already know about the SBN, we can predict some of the key genes that, that have already been described will be differentially expressed. So one of the key hormone receptors is androgen receptor, which is a target for testosterone. And this plot recapitulates this gene expression landscape across this single gene, showing that androgen receptor is not associated with mean T across much of the network, except in medial preoptic area. And so what is happening here is that as testosterone increases, the sensitivity of this region to the signal through the androgen receptor aspect of the androgen signaling pathway is increasing. And so the action of testosterone in this brain region is well described and seems to be a key component in aggressive behaviors. And so the result that's hot off the press from this week is that VRP receptor 1 and 2 are both downregulated in the pituitary, the time spent with social partners. These regulate vasoactive intestinal peptide in birds. And interestingly, VIP activity in the turkey pituitary stimulates the release of prolactin, which is also differentially but downly expressed with respect to social network strength. So this is super exciting because our knowledge so far about prolactin has only been linked with parental behaviours and migratory behaviours. So this is a new result, but it's super, super exciting. So far I have found all of these genes displayed here to be associated with social behaviour or a hormonal profile to be differentially regulated at some point in the network. However, looking at individual genes can only take us so far with all of the possible relationships involved. So we have a lot of work to do still in piecing this together. So I hope today what you have got from this is that this is a really, really cool study, and it's the first of its kind in wild birds. We have this unprecedented resolution of behavior and tissues and gene expression, and looking at this really unusual behavior. And I hope that I've highlighted that this is a really powerful data set with great potential, and also shown some starting results showing that we have differential expression in our key neuropeptides and hormone receptors that we already know are meant to be involved in social behavior. We've also shown system-wide gene expression landscape changes according to hormonal or behavioral trait. And any questions?